So let us now start with the discussion of the proteins. What you have to know when we are talking about proteins, so what part is getting asked in your exam. For proteins, you have to know the four orders of the protein structure, how the protein structure is organized. Okay, so primary, secondary, tertiary, quaternary, that in that way the protein structure is there. So primary structure, what is primary structure? It is just the sequence of amino acid in a polypeptide chain, and here the bond which is involved is the peptide bond. Please note, peptide bond is a very stable covalent bond, very stable covalent bond, which does not break easily. So when you do the denaturation of the protein, the primary structure will remain intact. So this question is commonly asked. The peptide bond is staying intact after denaturation. When you do the denaturation, the peptide bond will remain intact. It will remain intact after the denaturation. Then we have the secondary structure, which is having the contiguous amino acid. They form the superstructure. So here we get the non-covalent bonds. The non-covalent bonds will form the secondary structure. That is why when you do the denaturation, they will break. After denaturation, the non-covalent bonds will break. Please keep in mind, the non-covalent bonds will break. In the tertiary structure, we have two things. We have, number one, the non-covalent bond. In addition to this, in addition to non-covalent bond, we also have the sulfide linkages, sulfide bonds, and we also have the, okay, so non-covalent bonds and the sulfide bonds. So when you do the denaturation, again majority of the bonds are non-covalent bonds, so they will also break, and tertiary structure is also lost when you do the denaturation. So I'll write the same thing. After denaturation, the tertiary structure will also break. Now, this primary, secondary, and tertiary structure is described for those proteins where we have only one polypeptide, a single polypeptide structure is there. So for that, tertiary structure will be last. But in some proteins, we have one, two, three, four, multiple polypeptide chains are moved around each other to give the final product. For example, hemoglobin. Hemoglobin has four polypeptides. LDH. LDH has four polypeptides. Creatine phosphokinase. It has two polypeptides. So we have proteins having more than one peptide. For that, tertiary structure is not enough, because tertiary structure will tell us only about one polypeptide. But now we have more than one polypeptide. For that, we require the quaternary structure. So in quaternary structure, you get the non-covalent bond. You get the non-covalent bond. You also get the sulfide bond. You also get the sulfide bond. In addition to this, you get what are known as cross links. Cross linking are covalent bonds between the side chains. The side chain have the functional group, so they can also bind together by forming a complex. So the cross linking can be happening between the side chains. But majority of the bonds in this structure will also still be the non-covalent bonds. So the same thing will apply here also. After denaturation, the quaternary structure will also break. So the secondary, tertiary, and quaternary structure, they will break on denaturation. They will break on denaturation, whereas the primary structure is intact on denaturation. So the two important questions which are coming: what type of bond in which type of level of protein structure is there? What is there in the primary structure? What is there in the secondary structure? What is there in the tertiary structure? What is there in the quaternary structure? That question is asked. Number one. Number two, after denaturation, which of the structures will remain intact? Which of the bonds will remain intact? So peptide bond, primary structure. These are the two which will remain intact. All other will get broken down. The covalent bonds like the sulfide bond, disulfide bond, this should be the disulfide bond, or the cross linkings, they can remain intact, but they are not enough to form the tertiary or the quaternary structure. So secondary, tertiary, quaternary structures, they will be degraded on the denaturation. Another question related to the protein structure is the folding of the protein. See, the final structure will be achieved by the folding of the protein. So the molecules which assist in this folding are auxiliary proteins. Auxiliary proteins will assist in folding for a newly formed protein. And this is another question which is asked: which of the following proteins is an auxiliary protein? Which of the following proteins is not an auxiliary protein? So like that, we'll get the questions. So please note down the auxiliary proteins. These are the chaperones. Chaperones include the chaperonins and the heat shock proteins, the protein disulfide isomerase, the proline cis trans isomerase, the calnexin and calreticulin, and the GRP94 and BIP. These are some of the names which you have to be familiar with. So when you get the question on auxiliary proteins, you should know that these are the ones which may be deficient. So when the folding is correct, we get the suitable conformation of the protein. The correct final shape is achieved. The correct final shape is achieved if the folding is correct. However, if there is a defect in the folding, then the conformation will not be correct, and therefore we get the condition known as conformational disorders. So here what will happen, the primary structure is absolutely okay. After the translation we have gotten the correct protein, but when we go to the folding part, that is where we are not able to do the folding properly. For example, I'll write some examples here. In scurvy, the vitamin C is not there, which is required for hydroxylation of proline and lysine in the collagen. So what will happen, the collagen cross-linking will not happen because of the hydrogen bonds. Collagen will be weak. So in scurvy, then the prion disease, all of you know the prion disease. What happens here, the malformed protein goes near a normal protein. The normal protein also gradually becomes malformed. The shape changes, it acquires the improper shape. And then another name in this list is the Alzheimer's disease. Okay, Alzheimer's disease is also an example of the conformational disorder. Okay, so these are 2 points which you have to remember. Number 1, which are the auxiliary proteins, and number 2, examples of the conformational disorders. Sometimes you can get question about the collagen, where the collagen is located in which of the tissues. So we'll highlight some of the important ones. Collagen 1 is present in most of the connective tissue. You can see it is the most abundant collagen. If nothing else is mentioned, then when we say collagen, we mean collagen type 1. Collagen type 2 commonly present in the cartilage in the vitreous humor. Collagen 3 is important. Why? Because it is extensible. So wherever the extensible connective tissue is required, for example in the lungs, for example in the blood vessels, for example in the skin, so wherever the extensible tissue is required, there the collagen type 3 will be there. But please note, in that tissue still the most common will be type 1. Most common will be type 1. In all the tissues, most common collagen will be type 1. And then the collagen type 4 is slightly different from 1, 2 and 3. 1, 2 and 3 are straight, rod like, they're fibrillar. Type 4 is more globular, and that is why it is present in the basement membrane. It is present in the basement membrane. Okay, collagen type 7 is present as it is involved in you can say attachment between the different layers. It is getting hold of the different layers of the connective tissue, particularly in the skin. So type 7 also you have to remember. Okay, so that was a little bit about the proteins, some important points which are being asked from proteins, two things which were asked, and a little bit about collagen which is asked. Okay, here I'll mention some disorders which are related to collagen. Collagen associated with osteogenesis imperfecta. Osteogenesis imperfecta, I'm sure you've heard about this, you'll read more about this in your orthopedics, the osteogenesis imperfecta. So here what we have the fragile bone. Very easily the bones will break, so we have what is known as osteogenesis imperfecta. Now in addition to osteogenesis imperfecta, the other conditions involving the defective collagen, other conditions involving the defective collagen, we have what is known as Ehlers-Danlos syndrome. Ehlers-Danlos syndrome. Then we have the Alport syndrome. In Alport syndrome, you have to remember very specifically that defective gene is collagen 4A5. The type of collagen which is defective is collagen type 4. The fifth fibril is defective, called 4A5. Then we have what is known as Menkes disease. Menkes disease. In Menkes disease, you have to remember the defect is in a transporter for copper, which is the ATP7A. ATP7B is defective in the Wilsons. In Menkes, ATP7A is defective. And lastly, we have the condition known as epidermolysis bullosa. Epidermolysis bullosa, where collagen type 7 is defective. So the questions which are asked here, in Alport's, which particular gene is defective, which particular collagen is defective? Both answers I told you. The gene is collagen 4A5. The uh, uh, the collagen which is defective is collagen type 4. Now in Menkes disease, what is genetic defect? Genetic defect is the ATP7A, which is the copper transporter. Because of this, the copper is not available in adequate amount, and which enzyme is getting affected? The answer is lysyl oxidase. So here you have two questions: gene, what does it do? And the second question is which enzyme is affected? The answer is the lysyl oxidase. Third question is, in epidermolysis bullosa, which collagen is defective? So answer is collagen 7, which is working at the junctions. So because of this, bulla formation will be very easy. The two layers can just slip over each other. So bulla formation will be easier in these patients. The bulla formation will be easier in these patients. That's about the collagen. Some very simple straightforward questions being asked on collagen. You should be able to choose the correct answer. Let's move on to the carbohydrates. So about carbohydrates, first we have to understand their nature. So in nature, we have multiple hydroxyl groups along with aldehyde or the keto group. So first part, how do you describe them? They have multiple hydroxyl groups. They have either aldehyde or the ketone. Either aldehyde or ketone is there. So based on their function groups, based on their size, we classify them as monosaccharide, oligosaccharide, and the polysaccharide. So monosaccharide, oligosaccharide, and polysaccharide. Disaccharide is a subtype of the oligosaccharide. So names or numbers should be simple. If you have only one functional domain, we call it monosaccharide. In oligosaccharide, we have 2 to 10 functional domains, and if you have greater than 10 functional domains, then it is the polysaccharide. Okay. Moving on, some common questions which are asked. Smallest carbohydrate of the, okay, aldose family. What do you mean by aldose family? When we say aldose family, the functional group is the aldehyde. Aldehyde is the functional group. So it is glyceraldehyde. Then, which is the smallest carbohydrate of the ketose family? So here the ketone group is present. Then we have the ketose family. Here the answer is dihydroxyacetone. The answer is dihydroxyacetone. These are two straightforward questions which you must be able to answer. So the polysaccharides are further subdivided. They are subdivided as the homopolysaccharide, and we'll see later they are divided as heteropolysaccharides. So homo and heteropolysaccharide name is self-explanatory. When the same unit is repeating again and again and again, we get the homopolysaccharide. When different units are there, we get the heteropolysaccharide. So if the repeating unit is glucose, then the molecule is called glucosan or glucan. On the other hand, if the building block is fructose, then we get the term fructosan or the fructan. Okay. Please make special note that chitin is a type of homopolysaccharide where the building block is N-acetyl glucosamine. So chitin is also a homopolysaccharide. Moving on to heteropolysaccharide, I told you multiple units are coming together to form the polysaccharide. The classical example is the glycose aminoglycans, but before we go there, you should know some other examples. Hemicellulose, lignin, pectin, gum. These are some of the important, you can say, heteropolysaccharides in addition to the glycose aminoglycans. In addition to the glycose aminoglycans. About glycose aminoglycan, they contain amino sugar attached to uronic acid. Uronic acid means oxidized monosaccharides. Sometimes they are attached to the protein molecule. Sometimes they are attached to the protein molecule to form a proteoglycan. Sometimes they are attached to a protein molecule to form a proteoglycan. Okay, so proteoglycan is also known as mucopolysaccharide. So please keep in mind, mucopolysaccharide is not the glycosaminoglycan. Mucopolysaccharide is not the glycosaminoglycan. Rather, it is the proteoglycan, which is called the mucopolysaccharide. Proteoglycan, which is called the mucopolysaccharide. Let's look at the important glycosaminoglycan that you should be knowing about. Glycosaminoglycan, hyaluronic acid. This is the most abundant because it is very widely distributed in most of the connective tissue, so it is most abundant, followed by the chondroitin sulfate. It is present in the cartilage and in the bone, so because of this their amount is very high. So hyaluronic acid, chondroitin sulfate, these are the two most common glycosaminoglycan. Then we have the keratin sulfate type 1 and 2, where we don't actually have the uronic acid. Then heparin sulfate, and dermatin sulfate. Okay, these are some of the not some of the these are the glycosaminoglycans, which are there. Seven types of glycosaminoglycans. They attach with the proteins to form the proteoglycan, also known as mucopolysaccharide. Please note, sometimes the glycosaminoglycans can accumulate. When will they accumulate? When there is defective degradation. When there is defective degradation, they will accumulate. When they accumulate, they give rise to the condition known as mucopolysaccharidosis, commonly written as MPS. MPS, that is the mucopolysaccharidosis. When the mucopolysaccharidosis accumulate in the body, they are not getting degraded. We get the condition called mucopolysaccharidosis. Some names which are the conditions which fall under mucopolysaccharidosis. Some important names. Hurler. Hurler is now called MPS type 1. Subtypes are there, 1H, 1HS and 1S. So MPS type 1 M is Hurlers. Hunters. Hunter's is MPS type 2, whereas this Marotulami is actually MPS type 6. In addition to this, we also have the condition called Sanfilippo. Sanfilippo is MPS type 3, and Marcus, which is the MPS type 4. There is no MPS type 5. MPS type 6, I already told you, is the Marotu-Lamy disease. Sometimes you may even get the name of condition called SLY. This is the MPS type 7, but generally we don't talk about SLY, not very common. Most important would be the MPS type 1, 2 and 6. For MPS, what are the common features? Common features will be mental retardation, autosomal recessive inheritance, and a very prominent feature, that is corneal clouding. Corneal clouding along with mental retardation and autosomal recessive inheritance pattern. Here, the Hunter is having an exception because it shows the X-linked recessive inheritance pattern, and also there is no corneal clouding here. There is no corneal clouding in patients of Hunter. Obviously, see, when he is going to go for hunting, he will need the clear eyes, right? So corneal clouding is not there in Hunters, whereas in others you will see the corneal clouding, mental retardation, and autosomal recessive inheritance pattern. For Hunters, it is X-linked recessive along with no corneal clouding. Then we have what is known as isomerism in carbohydrates. When you talk about isomerism in carbohydrates, isomerism can be because of various factors. First, we have what is known as chiral carbon, means carbon where 4 valencies are occupied by 4 groups. This gives rise to the phenomena called stereoisomerism. The chiral carbon gives rise to what is known as stereoisomerism. In stereoisomerism, we see three things. Number one, enantiomerism. Number one, enantiomerism, where we have the D and the L forms. Okay, so stereoisomerism, first subtype is enantiomerism. Number two, diastereomerism. In diastereomerism, a very important subtype is epimerism. We'll come to this shortly. Let me first complete. So a very important subtype is epimerism. And third is anomerism. Anomerism, where we have the alpha and the beta forms, where we have the alpha and the beta forms. Then we can have isomerism on the basis of the ring structure. On the basis of ring structure, we can have two forms. We can have the pyranose form and we can have the furanose form. So for this, I'll tell you only one thing. Pyranose has a 6-membered ring, 5 carbon, 1 oxygen, that is 6-membered ring, whereas furanose has the 5-membered ring. How to remember? How to remember this? F for furanose, F for 5, F for furanose, F for 5. So 5-membered ring for furanose, 6-membered ring for the pyranose. Please note, anomerism is seen only in the ring structure. In linear structure, the anomerism is not seen. And last is the functional isomerism. Last is functional isomerism. So here you already mentioned what are the functional isomerism forms. You have the aldose and the ketose forms, right? We have said in aldose we have the aldehyde as the functional group, and in ketose we have the ketone group as the functional group. So these are the isomerisms which are seen in carbohydrates. Coming back to epimerism, what you have to remember: glucose and mannose. Similarly, glucose and galactose. These are epimers of each other. Glucose and mannose are epimers at carbon 2. Glucose and galactose are epimers at carbon 4. How to remember? How to remember? That is GM2, GM2 and GG4. How to remember? GM2 and the GG4. Glucose galactose will be there at carbon 4 and glucose and mannose at carbon 2. Okay, let's move on to the metabolism now. Metabolism of carbohydrate. I think the screen is stuck. Can we see this? Let me see. It's not moving forward. It's not moving forward. The screen is stuck. Okay, let me just quickly restart the screen. Let me quickly restart the screen. Close it and then I'll quickly restart it. It's connected but was not moving forward. It got stuck. Okay. Again it is stuck. Okay, so let us now talk about the metabolic pathway known as a glycolysis. Very important, lots of questions coming from glycolysis which you should know about. Let's see. Glycolysis, also known as the Embden-Meyerhof-Parnas pathway, written as EMP pathway. This is the Embden-Meyerhof-Parnas pathway. It is divided into two phases. One is the preparatory phase in which ATP is consumed. The second is the payoff phase in which the ATP is produced. So just like any other business, first you have to invest the money and then only you will start making the profits. So the investment is in terms of ATP and obviously the payoff is also in terms of the ATP. So very briefly, what happens here? The glucose or the glycogen acting as source gives us the hexose phosphate, right? Hexose phosphate in the form of glucose 6 phosphate is there. This hexose phosphate will be split into, so we have two trioses which are interconvertible. You can see they are interconvertible, and then one of the trioses is metabolized right up till the pyruvate. One of the trioses metabolized till pyruvate. Normally, this pyruvate has to go to mitochondria where it undergoes further metabolism in aerobic condition. But if anaerobic condition is there, then the pyruvate will go to lactate. So pyruvate to lactate, this is happening only in anaerobic condition. Normally it doesn't happen. It will happen only in the anaerobic condition. Okay. You also see that normally the NADH is generated, but when you convert pyruvate to lactate, this NADH is used up. In addition to this, during the conversion of triose phosphate to pyruvate, two ATP are formed, and during this preparatory phase, two ATP are consumed. Two ATP are consumed. Okay, so what happens? We have two trioses which have to get metabolized. So this production of 2 ATP occurs twice, giving us the 4 ATP. We get the 4 ATP. and you can see only 2 ATP have been used up in preparatory phase. So when you do the anaerobic glycolysis, we are just measuring the ATP which is being generated in the glycolysis itself. We get the 2 ATP. This is from anaerobic glycolysis, right? This is from anaerobic glycolysis. But if you have the aerobic glycolysis, then you will have NADH also, and this will also be present in twice the amount. This will also be present in twice the amount. So depending on which shuttle you are using, there are two options which you can use. You can either use the malate aspartate shuttle. You can use the malate aspartate shuttle, or you can use the glycerol phosphate shuttle. If you use the glycerol phosphate shuttle, the NADH is getting converted first to FADH2 and then it releases the energy. So you will only get 1.5 ATP per NADH, resulting in formation of 3 ATP. Whereas if you go through the malate aspartate shuttle, you will get 2.5 ATP because NADH will remain intact, and ultimately you get the 5 ATP. Okay. All right. To this you have to add. To this you have to add this number. So depending on the shuttle which is used in aerobic glycolysis, you either get 5 or 7 ATP. You either get 5 or 7 ATP. This is in aerobic glycolysis. If you are making use of glycogen, in that case, in that case, only one ATP is required in activation, because conversion of glycogen to the glucose 6 phosphate does not require ATP. So with glycogen, your net gain should be 3 ATP. For glycogen, your net gain should be 3 ATP. Very important question. It has been asked quite a few times recently. So when glycogen is fuel for anaerobic glycolysis, we get the 3 ATP. And lastly, you have to remember that in RBC, we have what is known as the Rappaport-Lewis shunt. In RBC, we have what is known as Rappaport-Lewis shunt, whereby 2,3-bisphosphoglycerate is synthesized. 2,3-bisphosphoglycerate is synthesized. As a result, if you're talking about glycolysis inside the RBC, if you're talking about the glycolysis inside the RBC, in that case, our gain in case of RBC, our gain should be 0 or 1 ATP. Our gain should be 0 or 1 ATP. Okay, all of this we've done. So what are the points you should know separately for glycolysis? Let's see the points which you have to know for glycolysis. Which are the tissues which are dependent on glycolysis? Tissues which are dependent on glycolysis. See, these are tissues which have exclusively anaerobic glycolysis. These are tissues which have exclusively anaerobic glycolysis. RBC doesn't have mitochondria, the others do not have the blood supply. Whereas retinal medulla and brain have aerobic plus anaerobic glycolysis. They have aerobic plus anaerobic glycolysis. The renal medulla and the brain have aerobic plus anaerobic glycolysis. Okay. So these are the tissues which are dependent on glycolysis. Second question which is commonly asked from glycolysis is Enzymes which catalyze the irreversible steps. Enzymes which catalyze the irreversible steps of glycolysis. These include the hexokinase, the phosphofructokinase, the pyruvate kinase. Okay, they catalyze the irreversible reaction. All other reactions of glycolysis are freely reversible. And we already said, due to the Rappaport loop bearing shunt, due to the Rappaport loop bearing shunt, the first site of ATP formation is bypassed. Now a little bit about regulation. Let's talk about the regulation. So what you have to remember, hormonal regulation acts on PFK2. How? By phosphorylation, dephosphorylation. In case of phosphorylation, it is inactive. When it is dephosphorylated, then it is active, meaning inactive form will be from the glucagon or even epinephrine, whereas active form will be due to the insulin. Okay, hormone regulation is acting via the PFK2 through the mechanism of phosphorylation and dephosphorylation. PFK1 is under allosteric regulation. Allosteric regulation, some of them are activators, some of them are inhibitors. The activators are AMP, fructose 6-phosphate and fructose 2,6-bisphosphate. AMP, fructose 6-phosphate and fructose 2,6-bisphosphate. Inhibitors are ATP and citrate. Inhibitors are ATP and citrate. Please note, PFK2 gives rise to fructose 2,6-bisphosphate, meaning this is an element of indirect hormonal regulation. You can think of the fructose 2,6-bisphosphate in this case to be acting as maybe second messenger, not exact, but you can think of it like that. So PFK1 is also indirectly under hormonal regulation. PFK2 is directly under hormonal regulation. PFK2 is indirectly under hormonal regulation via the amount of fructose 2,6-bisphosphate, which is the product of PFK2. Then we have the enzyme hexokinase. Hexokinase will show feedback inhibition by the glucose 6 phosphate. It will cause feedback. It is showing feedback inhibition by the action of glucose 6 phosphate. Here, what you have to note, for the glucokinase, the concentration is increased by insulin. So glucokinase is also affected by the hormones. Glucokinase is also affected by the hormones. On the other hand, hexokinase is not affected by hormones. It is not affected by hormones. Okay, this question is asked quite frequently. Which of the enzymes of glycolysis are not affected by hormones? So four options will be there. So in that case, you have to remember, if hexokinase is there in the option, you have to mark hexokinase. Hexokinase is not affected by hormones. Okay. So at the end of glycolysis, you get the pyruvate. The pyruvate can be further metabolized in the cytoplasm. In the cytoplasm, as we've already seen, it can be converted into the lactate. This reaction is reversible. It can also be converted into alanine by transamination. By transamination, pyruvate can be converted into the alanine enzyme being alanine transaminase, whereas for lactate, the enzyme is lactate dehydrogenase. This is occurring in the cytoplasm. The pyruvate can also go to the mitochondria, and in mitochondria it can either be converted into oxaloacetate. Please note that the reaction is irreversible, or it can be converted into the acetyl coenzyme A. Again, note the reaction is irreversible. For acetyl coenzyme A, we have the pyruvate dehydrogenase complex, whereas for oxaloacetate we have the pyruvate carboxylase. Carboxylation is required because The auxiliary state has 4 carbon, whereas pyruvate only has 3 carbon. So we require the carboxylation step. So these are the fates of the pyruvate. This acetyl coenzyme will go to the Krebs cycle, whereas auxiliary state will go for the process called gluconeogenesis. It will go for the process called gluconeogenesis. New glucose will be formed via this mechanism. Little bit about the pyruvate dehydrogenase complex, which converts the pyruvate to acetyl coenzyme. The points which you have to keep in mind: it has 3 enzymes and 5 coenzymes. It has 3 enzymes and 5 coenzymes. The coenzymes are derived from vitamins. Most of them derived from vitamins. The TPP is derived from vitamin B1, FAD is derived from vitamin B2, NAD is derived from vitamin B3. Coenzyme A is derived from the pantothenic acid, which is also written as B5. Also note, the pyruvate dehydrogenase complex is similar to 2 other complexes. These include the alpha ketoglutarate dehydrogenase complex and the branched chain keto acid dehydrogenase complex. These are the 2 enzyme complexes which are similar to the pyruvate dehydrogenase. Means each of them will have the 3 enzymes, second and third are common. Each of them will have the 5 coenzymes. If any of the coenzymes is missing, then the whole complex will stop working. Pyruvate can be inhibited by certain conditions. One is presence of arsenite and mercuric ion. Second is the dietary deficiency of thiamine. You already seen thiamine is required in the form of thiamine pyrophosphate. All right. So if thiamine is not there, then the thiamine pyrophosphate will not be there, and the pyruvate dehydrogenase will stop working. Lastly, in some individuals there is inherited pyruvate dehydrogenase deficiency. In some individuals there is inherited pyruvate dehydrogenase deficiency. Now we come to the Krebs cycle. I'll not go into the Krebs cycle proper. What you have to understand is the calculation in Krebs cycle. 10 ATP are generated per turn, or you can say per mole of acetyl coenzyme. You get the 10 ATP. How do you get 10 ATP? There are 3 NADH. Each NADH will give 2.5 ATP, giving you a total of 7.5. There is one FADH2 which will give rise to 1.5 ATP, and one substrate level phosphorylation. When the ATP is directly generated during the reaction, we call it the substrate level phosphorylation. Okay. If we start from pyruvate instead of acetyl coenzyme, if we start from pyruvate, when the pyruvate is converted into the acetyl coenzyme, 2.5 ATP are formed. So if you start from pyruvate, 12.5 ATP. If you start from acetyl coenzyme, in that case you get only 10 ATP. So that is briefly about the Krebs cycle. Very briefly about the Krebs cycle. One extra point which I wanted to know here about the Krebs cycle. Here the rate limiting enzyme is isocitrate dehydrogenase. The rate limiting enzyme is isocitrate dehydrogenase. And second point, the acetyl coenzyme will never contribute to gluconeogenesis. This is another common question which is asked, and one of the options is acetyl coenzyme. So remember, the acetyl coenzyme will never contribute to gluconeogenesis. Why? Because along with these energy products that we say, we also get 2 carbon dioxide. Due to the decarboxylation, 2 carbon dioxide is released. So acetyl coenzyme has 2 carbon. During the Krebs cycle, you have removed 2 carbon. So there is no net gain. So it cannot contribute to the gluconeogenesis. So this is the important point which you have to keep in mind. Talk about gluconeogenesis, few things which you should note, which are the substrates. Very common, very important, repeatedly asked. Glucogenic amino acid, lactate, pyruvate, propionate, glycerol. Which is the most important substrate for gluconeogenesis? The answer is alanine. The answer is alanine. Lactate is the most common substrate or source for gluconeogenesis. After strenuous exercise, means when you do the very severe exercise, only then a lot of lactate is accumulated. This lactate will go to the liver and it will act as a fuel for gluconeogenesis. So after strenuous exercise, lactate will become the most important or most common substrate. Then we have pyruvate, propionate. Propionate is coming from the odd chain fatty acid. Please keep in mind the last 3 carbon of odd chain fatty acid will get converted to propionate. From there it will go to propyl coenzyme, then succinyl coenzyme, and then the Krebs cycle. Okay, so these are the substrates which are involved in gluconeogenesis. Second, you should know which organs will show the gluconeogenesis. So here your answer is liver and kidney. Compartments which are involved are both cytoplasm and mitochondria. Both cytoplasm and mitochondria will be involved in gluconeogenesis. Enzymes which are unique to gluconeogenesis. See, we said the glycolysis, most of the reactions are fully reversible, but some of the reactions are catalyzed by some of the irreversible reactions are there, which are catalyzed by specific enzymes. So here, what you have to note: pyruvate carboxylase and pep carboxykinase are going to bypass the activity of pyruvate kinase. The fructose 1,6-diphosphatase will bypass the activity of phosphofructokinase 1, whereas glucose 6-phosphatase will bypass the activity of hexokinase. So please make note of these two very commonly asked which enzymes in the uh, gluconeogenesis with enzymes in glycolysis. These are the enzymes of glycolysis and they are bypassed by the respective enzymes when we are doing the gluconeogenesis. Let us quickly look at the questions. Some questions. Screen is getting stuck again and again and again. Okay. Glycolytic enzyme inhibited by fluoride. Very important common question. Answer is enolase. The second last enzyme of glycolysis inhibited by fluoride. The answer is enolase. The answer is enolase. Screen is stuck. A lot of problem with the software today. Okay. Question number 2. 3-ATP in anaerobic glycolysis occurs with which fuel? We just said 3-ATP in anaerobic glycolysis occurs with glycogen. So correct answer is glycogen. Next question. Anticoagulant used to estimate glucose from sample sent for PSC. Please note, you are not going to mark sodium fluoride here. You are going to mark sodium fluoride with a strong anticoagulant. See, the question itself says you anticoagulant used to estimate glucose. So you have to mention the anticoagulant along with sodium fluoride. It must be attached to sodium fluoride. So it can either be potassium oxalate or it may even be the EDTA itself. Any of the two can be there attached to the sodium fluoride. Sodium fluoride must, because it will block the glycolysis, but along with sodium fluoride you must have one strong anticoagulant, whether it is potassium oxalate or it is EDTA, that's up to you. After overnight fasting, level of glucose transporter are reduced in, please note the answer here will be adipocyte and muscles due to the GLUT4, which is dependent, sorry, GLUT4, not like this, so write that. GLUT4, which is dependent on insulin. So when there is overnight fasting, the insulin level will fall. The GLUT4 goes inside the cell. Once it is inside the cell, it cannot take up the glucose. So adipocytes and muscle, which are made up of the glut, which are having the GLUT4 on their surface in presence of insulin. When the overnight fasting is there, the transporters will go inside. So the level of glucose transporter will be reduced. The level of glucose transporter will be reduced. So you already done this. Pyruvate can be converted directly to all of the following except can be converted to alanine, alanine transaminase. Oh, screen is not changed. It is still stuck, not going ahead. It's a big problem. Come on, come on. Screen is getting stuck again and again. Let's see. Okay, let's go. Question number 2. Pyruvate can be converted directly to all the following except we've already done this. It can be converted to alanine by the alanine transaminase, acetyl coenzyme by pyruvate dehydrogenase, lactate by lactate dehydrogenase. No enzyme for synthesizing the PEP directly. No enzyme for converting it to PEP directly. Arsenite inhibits all except here you have to make note. Arsenite will inhibit the PDH. It actually inhibits the lipoic acid, which is also present in the branch chain keto acid dehydrogenase. So it doesn't act on isocitrate dehydrogenase. So arsenite will inhibit lipoic acid. The lipoic acid is present in the PTH and the branch chain keto acid. So these will also get inhibited. Congenital lactic acidosis is due to defect in pyruvate dehydrogenase. This is the inherited deficiency. We had said inherited deficiency of pyruvate dehydrogenase will cause the lactic acidosis. In vivo control of citric acid cycle is affected by ATP. Please note this down. You can write separately. This is a commonly asked question which is not mentioned in a lot of books. Cytosolic cytochrome C mediates, please keep in mind, apoptosis. If it is in the mitochondria, then it can participate in the electron transport. But outside the mitochondria and the cytoplasm, it will induce the apoptosis. Which of the following sugars are both non-reducing? Please note this answer. Sucrose and trehalose are both non-reducing. Okay. The maltose and lactose, these are reducing disaccharides. Okay, glucosamine are used in the treatment of arthritis. Glucosamines are used in the treatment of arthritis. Following tests are done for protein. This is for protein, this is for sugar, and this is for ketones. Which are the tests? First test is the biuret test. This is for protein. Second test is the Benedict's test. This is for reducing substance. And third test is the Rothera's test, which is for the ketone bodies. So now when you have the starving patients, which are the following will be positive in starvation state in urines, only the ketone bodies will be there, which is shown by the Rothera's test. So your answer should be only 3. Rothera's test will be positive in patients of starvation, because here they will be having the ketosis. Moving on, most important rate limiting enzyme of glycolysis. So again you can mark it here, PFK1. Most important rate limiting enzyme of glycolysis is PFK1. Okay, please note PFK1 is not directly, it is not directly affected by the hormones, it is only indirectly affected by the hormones. Okay, let's really talk about glycogen. What you should know about glycogen: it is the storage form. It is the storage form in animals, and it is a branched polymer. It occurs mainly in liver and muscle. Mainly in the liver and muscle. So two things you have to remember: the glycogen inside the muscle, it is for local use within the muscle itself, whereas the liver glycogen is used to maintain the blood glucose. The liver glycogen is used to maintain the blood glucose. So glycogen metabolism, uh, it is a story of storage and utilization. So in the fed state we make the glycogen, and in the fasting state we reutilize the glycogen, or when we are under stress, when we do a lot of exercise, physical activity, so that energy can come from the stored glycogen. Sometimes the synthesis and breakdown may be defective, giving rise to the condition known as glycogen storage disorder. So you can see the name is there, and now the type is there: 1, 2, 3, 4, 5, like that we have the types. So what remember, number one, von Gerke disease. What is a defective enzyme? Enzyme is glucose 6 phosphatase. For Cory's disease, which is the defective enzyme? Defective enzyme is the D branching enzyme. In Cory's, the D branching enzyme is defective. What is happening in Anderson's disease? The branching enzyme is defective. In Anderson's, the branching enzyme is defective. What about McArdle's? The myophosphorylase is defective. In McArdle's, the myophosphorylase is defective. And in hers, the hepatic phosphorylase is defective. In hers, it is the hepatic phosphorylase which is defective. So with these tools, you can remember them easily. In Cory's, D branching, C followed by D. In Anderson's, we have the B branching, A followed by B. M for M in McArdle, myophosphorylase. H for H in hers, it is the hepatic phosphorylase. And lastly, we go to the electron transport chain. Lastly, we go to the electron transport chain. Electron transport chain, you have to make note of the complexes and their names. Here you have to make note of complexes and their names. Please note, each of the complexes 1, 2 and 3 contain FES, but not in complex 4. Each of the complexes 1, 2 and 3 contain the FES, but not in complex 4. In addition, you can see the presence of heme in complex 4, which is not there in complex 1, 2 and 3. Presence of heme, which is not there in complex 1, 2 and 3. And secondary, the presence of copper, again not present in 1, 2 and 3. It is not present only in complex 4. So what happens? Each of these complex 1, 3 and 4 will pump out protons. It will pump out protons. so that from the matrix in the intermembrane space you find more of the proton is present in the intermembrane space, so it wants to go back to the matrix. But you remember, the inner material membrane is impermeable, nothing can cross the inner material membrane, so it requires channels. And one example of that channel is the F0 F1, also known as ATP synthase, also known as complex 5 of the electron transport chain. So you can see the F0 subunit and the F1 subunit. So protons will pass through the F0 subunit, they cause the rotation. This rotation is transferred to the F1 subunit, which will bring the ATP and ADP together to form the ATP. So this is where the phosphorylation is taking place. This is where the phosphorylation is taking place. In complex 1, 2, 3 and 4 it was the oxidation. Electron is being used up, so that is the oxidation. So we have the oxidative phosphorylation. Now you should know the inhibitors of electron transport chain, very commonly asked. Inhibitors of complex 1: Pecitin A, amobarbitol, amitol, the rotenone, the secobarbitol, the chlorpromazine, the guanethidine, demerol. All of these are inhibitors of complex 1. Okay. In complex 2, we have the inhibitors called malonate and carboxyl. Malonate and carboxyl will inhibit complex 2, whereas in complex 3, we have the fenformin. Fenformin is there in complex 3. But by far for our exam, the most important inhibitors are for complex 4. Most important inhibitors for complex 4, which are those: the hydrogen sulfide, the carbon monoxide, the azide and cyanide. Hydrogen sulfide, carbon monoxide, cyanide and azide. These are the 4 inhibitors of complex 4. Almost always you will get the question from these. You can also get the question on inhibitor of complex 5. We told you in complex 5, the ATP synthase complex, also known as the F0 F1 complex, right? So here the oligomycin is inhibitor of F0 subunit. And another inhibitor you need to know is for the ATP ADP translocase. The answer in this case is atractyloside. This question has been asked a few times. You should know these points. Lastly, we have the molecules known as uncouplers. What they do? The electron transport chain will keep working normally, complex 1, 2, 3, 4 working. When it is a story for the F0 F1 complex to work, they will take the protons through some other channel so that they do not enter the F0 F1 complex. We call them the uncouplers. So which are the uncouplers? The exogenous uncouplers are phenolic compounds and dicumarol. The exogenous uncouplers are phenolic compounds and dicumarol. The endogenous uncouplers are receptor specific. UCP1 molecule acting is thermogen. UCP3 commonly distributed all over the body, acted upon by a large number of hormones, particularly the catecholamines and the thyroxine, norepinephrine, epinephrine. Sometimes even leptin can be there. For the UCP4, absence of the essential fatty acid, that can be one problem, and second is the presence of purine nucleotides. So these are the endogenous uncouplers. They are important to maintain the body heat. Even if you are sitting in an air conditioned room, your body temperature is maintained. How? It is with the help of the uncouplers. They generate the heat in the body. Some of the energy is liberated as heat. It will not be there in the cold-blooded animals. There, these uncouplers are not required. Okay, please take a short 10-minute break, and then we finish the topic of lipids and the vitamins. Short break, 10 minutes, and then we complete it.